Hi guys, Dr. H here. This short video is just going to be a brief overview of DNA and chromosomes and how those chromosomes are moving around in the cell. So this is not going to go in depth into DNA sequence or DNA mutations or those kinds of heredity things. We're going to talk about that later on in the next unit. This is just the information that you need to know in order to understand mitosis. So let's just start off here by having a review of what DNA is. So remember we talked about in classes, the DNA is what holds all of our cells' genetic information. They are the instructions for making all the proteins that give you the characteristics. It's what makes you unique. Everybody has their own special sequence. They're going to be similar, but they're going to be a little bit different. And that's what makes you unique and awesome, because it's your uniqueness that makes you beautiful. And everybody is beautiful in your own special way. And I really believe that. And always remember that, seriously. Okay, so DNA, where is it found? It's going to be found in the nucleus of your cell. And remember, the nucleus is what's going to be divided during mitosis. We will divide the cytoplasm later in cytokinesis. But right now, we're going to focus on nucleus and DNA because that's what's divided during mitosis. So let's just kind of look at the numbers of DNA. This is kind of like astounding. I think this is so crazy cool to imagine. So think about this, that each of your cells have about 2 meters of DNA. That is like bigger than I can hold my arms. Think of those 2 meter sticks together. That's how much DNA is packed in one single tiny little cell that's in your body. Remember we looked into those at the cells under the microscope? Super tiny. And you get 2 meters of DNA packed in each one of those? That's crazy cool. And think about this even further. Is your average human has about 75 trillion cells. So if you do the math, that is 150 trillion meters of DNA in your entire body. And that is enough DNA that you can go from the earth to the sun between 400 and 600 times. That is crazy cool. All of that in your body. That is so neat. Isn't science awesome? Yes. So how does it get packed into there? Well, here's the crazy cool thing again, is that the DNA is so thin. It's so tiny. And it has a diameter that's only, let's look at these zeros because I have to count them out. 0.000000002 meters. That's how tiny thin it is. That's insanely thin. So that's why it can get all packed into that nucleus into where you still can't see it because you can't even see cells. So all of that has to be packed in. Now think about this. As we talked about, DNA is a long line single strand. So you think about DNA kind of looks like this double helices, and again, we'll go into the structure later, but typically it's like a long strand of yarn. And you've got like 46 of them because you have 46 tiny pieces of them. There are 23 from your mom, 23 from your dad. And those things are typically wound up inside the cell, packed into like a little ball. This is what it looks like during interphase. It's all packed up into a tight little ball, all wound up tightly packaged together, so it can fit into that nucleus. So the thing is that if we've already replicated that. So not only do we have 46 long strands that are all wound up, now we have 92 of them because we had to duplicate them because we have to split them during mitosis. Remember, each daughter cell needs to be an exact copy of the parent. So we have to make sure we duplicate the DNA before we can split it so it's identical. Okay, so that's going to be like a hot mess to try and like split like 46 individual like 2 meter long from another 46, and then how are they going to get all mixed up? It's going to be like a hot mess. And you know, like Dr. H, I don't like disorganization. I don't like messes. It's like, it kind of freaks me out. Same thing here. The cell does not like to be disorganized. Remember, that's when errors can happen, and we can't handle errors because that causes mutations. Not the good kind of mutations that we really want, like X-Men. Like, how cool would that be? No, not those kinds of mutations. So we need to make sure that it's very organized, and the cell has a way to keep that organization. So what it does is it takes that ball of DNA that's found in interphase, and then it starts to separate out each of those 46 chromosomes, and they start to condense down into what are called chromosomes. So those chromosomes are just condensed DNA, just where they're all tightly wound and structured to where it's going to be easier to split up. So we got 46 copied versions of them. So let's just look at this too, is that one single chromosome, remember we have 46 of these single chromatids, but we've already duplicated them, so we've got 2 matching identical ones of that. Now we've got 2 identical chromosomes here, But if we want to make sure that they split up evenly, and we want to make sure that they don't like split up like the same duplicate goes on one side and then the other we got to keep them together so that we can kind of organize them. And this really cool thing called a centromere comes into play here. So the centromere is kind of like a little tube or like a protein that comes in and like sticks those two together, and it contains them and they stick together. Those duplicated chromosomes stick together until they need to be pulled apart so that they can move as one unit. So a centromere here, and these two duplicated chromosomes are what we call sister chromatids. I don't know why we don't call them brother chromatids. I mean, that's kind of weird, but I don't know. Anyway, but we have sister chromatids. These two held together by a centromere. In my mind, I have this like little like crazy analogy. I think of these two little cute little 5-year-old identical twin girls, and they're just like la 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 hanging around and playing. You know how 5-year-olds have like so much energy, they're like running around everywhere. Gotta keep them together. So like think about this: if you're taking them to a pool party, they're gonna go to a pool party, they're gonna jump in the pool, and they're gonna like want to play together. If you've ever like played with your friend and like tried to get in the inner tube, like one of those cool little round inner tubes, and where you try to like bounce another person off, but you're like stuck in together and trying to stay together, I kind of think these two little twin girls like hanging out in an inner tube holding them together. They move as one little unit. I think it's kind of cheesy, but it'll come into play a little bit better later on. Anyway, that's what I think in my mind. Okay, so how are we going to move those like two sister chromatids around in the cell, move them where they need to go when they need to be there? So that's where these really cool things come into play called centrioles. Now these guys are like super stealthy. So like they only appear during division. You don't see them in the cell unless the cell is dividing. Now these cool little protein organelles, they are going to kind of originate, and they're going to make sure that they locate themselves at the edges of a cell. So they're going to be on opposite sides of a cell or opposite poles. Think of like the north and south pole on the earth. These are on opposite poles of the cell because that's going to be like your points where they're going to pull the cell apart. Those chromosomes are going to be pulled to them. So I always remember that centriole rhymes with pole. So that might help you remember what it does and which one it is. So don't confuse it with centromere. Centriole goes to the poles. Ah, yeah, I got some ways to remember it. Okay, so they're going to go to the edges of the pole. They're going to have these really cool things where they stick out and they shoot out these fibers that are going to grab on to those centromeres and pull those chromosomes where they need to go. So you can see here in our drawing we have these centrioles, and they're shooting out spindle fibers that will latch on to those centromeres. Now, this is where my analogy kind of gets kind of crazy, like as if it wasn't already, but I kind of think about those two little girls at the pool party, and these centrioles are like Spider-Man lifeguards. They're like, I'm going to sit here and I'm going to monitor the entire pool party. I'm going to make sure everybody's safe. And those two twin little lifeguard girls are like, woo, like floating around the pool, and they're going to the deep end where they don't need to go, and they're like, ah. And those Spider-Man lifeguards can be like, whoa, that's not where you're supposed to be. I'm going to take you where you need to go. And they're going to like shoot out that like webbing and like snatch onto the inner tube and pull them where they need to go, pull them back into safety. That's kind of what these centromeres or centrioles are doing. They're standing at the edge, shooting out those spindle fibers that are going to grab onto the centromeres and pull them where they need to go. Okay, so if that analogy helps, I mean, maybe you can use it. If it doesn't help, then whatever. It's just a cute analogy. You can make fun of it all you want, just not out loud because mean people suck. We're going to be nice. We're going to build people up, not tear people down. Let's remember that. Let's be good for society, okay? All right, so again, like always, if you have questions, come see me before and after school. Send me an email. Shoot me a message on Edmodo. Let me help you out. I want to make sure you study all this information. I want to make sure that you understand it. I want to see you do really, really well and learn. Be awesome. Keep studying. I will see you soon. Bye.